Hey everyone, welcome to today's Explainer. Today we're going to take a journey through time to uncover the incredibly fascinating history of cell theory. We'll be looking at exactly how humanity managed to unlock a completely invisible, microscopic world that had been hiding right under our noses for literally millennia. We're going to meet some brilliant pioneers, check out their ingenious inventions, and really get into the fundamental rules that govern absolutely everything alive today. Let's get into it.
All right, let's kick things off with a bit of a mystery. I want you to think about this for a second. What could a towering, massive redwood tree, a completely microscopic bacteria, and you, a highly complex human being, possibly share in common? I mean, on the surface, it looks like absolutely nothing, right?
But the answer? You're all built from cells. Being made of cells is the absolute primary characteristic of all organisms. It literally doesn't matter if we're talking about a plant, an animal, or a bacteria. Cells are the most basic fundamental unit of life. They are the ultimate building blocks. But, for most of human history, we had zero idea they even existed, simply because we couldn't see them.
To understand how we finally cracked this microscopic world, we're going to quickly map out four major breakthroughs: Robert Hooke's initial observations in 1665, Leeuwenhoek spotting living cells in 1673, Schleiden formalizing plant cells in 1838, and a massive modern twist from Margulis in 1970.
Starting with Section 1, The Pioneers, exploring the 1600s microscopic frontier. We are heading back in time to the brilliant minds who first figured out how to peer into the unknown.
Okay, picture this. The year is 1665. An English scientist named Robert Hooke is utilizing this elaborate early microscope to illuminate and examine a completely hidden landscape that nobody had ever properly documented before.
Hooke decides to look at a super thin slice of everyday cork, and when he does, he observes these incredibly strange, tiny, box-like structures. Now, what he was actually looking at were the dead cell walls of a plant. Since cork comes from tree bark, the living parts of the cell were long gone, leaving just this rigid, grid-like skeleton behind.
And here's a fun fact. Hooke actually coined the term cells for a pretty quirky reason. Those empty, rigid little boxes he saw in the cork simply reminded him of the sparse, unadorned quarters that monks lived in at monasteries, which were literally called cells. And, well, the name stuck.
Fast forward just a few years to 1673. We've got a Dutch microscope maker named Anton van Leeuwenhoek taking the next massive leap. Using these tiny, simple handheld microscopes he built himself, he bypassed the deadwood and pointed his lenses at pond water, and even scrapings right off his own teeth. Gross but revolutionary.
This perfectly illustrates the massive leap in scientific discovery happening here. While Hooke was observing the empty, rigid walls of dead plants, Leeuwenhoek was the very first person in history to view actual, living, moving microscopic organisms. The guy was literally watching the chaotic dance of life in a single drop of water.
Moving on to Section 2, the unification, formalizing cell theory in the 1800s. Let's see how these neat, isolated observations turned into a unified biological law.
Meet Matthias Schleiden. In 1838, this German botanist spent his time rigorously analyzing all of those previous microscopic discoveries alongside his own massive amount of plant research. He wasn't just looking at random samples anymore, he was hunting for a pattern.
And based on those observations, Schleiden officially concluded that every single plant, no matter its shape, size, or species, was made of cells. This was an absolute game changer. He essentially unified the science, proving that cells weren't just some quirky feature of cork, but the universal building block of all plant life. That made him a co-founder of what we now call cell theory.
Which brings us to Section 3, the modern twist, the 1970s endosymbiotic theory. We are skipping ahead over a century here, looking deeply at what's actually inside the cell.
So, the real crucial point here comes from American biologist Lynn Margulis. In 1970, she dropped the endosymbiotic theory on the scientific world. Margulis provided incredible evidence that some of the complex parts inside our cells, called organelles, were at one time ancient, free-living bacterial cells themselves. I mean, how did she prove that? She pointed out that specific organelles, like mitochondria, the power plants of the cell, and chloroplasts in plants, actually have their very own DNA, totally separate from the main cell nucleus. Basically, over billions of years, these tiny free-living cells moved inside larger cells, and they just set up shop and stayed.
Now for Section 4, the reality of cells today, and why cell size matters. Now that we know how they were discovered, let's look at the physical reality of these biological building blocks.
Have you ever wondered why we're made of trillions of tiny cells rather than just like a few giant ones? Well, it all comes down to simple math. As a cell grows, its internal volume increases way faster than its outside surface area. Eventually, that outer membrane simply becomes too small to effectively pump out the waste the cell produces or to pull in enough food and water to keep the inside alive. So, to survive this geometric crisis, the cell absolutely has to divide. That is exactly what keeps cells universally tiny and highly efficient.
Because every cell across all living things is strictly limited by these exact same physical laws, it leads us to a really fascinating question to wrap up our explainer today. Think about this. Are the cells inside a massive, lumbering elephant bigger, smaller, or about the exact same size as the cells inside a tiny little field mouse?
Surprisingly, they are basically the exact same size. The giant elephant isn't made of giant cells. It's just made of many, many, many more standard-sized cells than the mouse. From Hooke's dead cork to Leeuwenhoek's living pond water, all the way to you and me, these microscopic blocks really are the great unifier of biology. Which leaves us with one final thought. If the engines powering our own cells are actually the ancient descendants of captured bacteria working in harmony, what other invisible microscopic partnerships might be secretly shaping the future of human evolution right now? Keep exploring, keep questioning, and I'll catch you next time.